Hello, everyone, and welcome to today's webinar, Fortifying Plants in Time of Stress Using Foliar Application, presented by Balchem Plant Nutrition. I'm Robin Sittberg of Meister Media Worldwide, publisher of American Fruit Grower Magazine, and I'll be moderating the webinar today. This webinar has been approved for a half CEU credit in nutrient management from CCA. To receive credit, you must watch the entire webinar. Your names will be submitted to CCA 2 weeks from this live airtime. We will have time for some questions at the end of the webinar, so if at any time during the presentation you'd like to ask a question, just go ahead and type it in the question pane at the lower left panel of your screen and click Submit. You can do this at any time during the webinar, but we'll answer questions at the end. Before we begin, I'd like to introduce today's speaker. Jeremy O'Brien leads Balchem Plant Nutrition's commercial business as the Global Director of Sales. He's been with Balchem and legacy companies since 1998. Prior to his current role, Jeremy has held many positions, including that as lead agronomist covering the Pacific Northwest, Southern United States, Midwest, South Africa, and Australia. This experience has given him the opportunity to hone his agronomy and botany skills and help growers overcome many challenges. He's also been able to research plant nutrition on dozens of crops. Jeremy resides in Utah and earned a BS from Weber State University. So take it away, Jeremy.
Okay, thank you, Robin. Good day to everyone, and a big thank you for joining this webinar today. As was mentioned, Jeremy O'Brien is my name, and I work for Balchem Plant Nutrition. I've worked in the plant nutrition industry for going on 25 years now, and I'm looking forward to the opportunity of spending this time today discussing how we can fortify plants in times of stress using foliar application of nutrients. There are two types of stress that affect plant growth and development. The first type is biotic stress, and the second type is abiotic stress. Under the biotic stressors, we see things such as bacteria, fungi, viruses, animals, and weeds. And under the abiotic stress category, we find such things as temperature, drought, rainfall, salinity, and UV light. When plants experience stress, it's typically more than one of the stressors that is affecting the plant at any given time. It is important to understand what types of stress the crop is experiencing to be able to address overcoming those stress factors. It is important to create healthy plants or to provide plants with all the things that they require to be healthy. Healthy plants are more able to overcome and resist stress both by abiotic and biotic factors. Balanced nutrition leads to healthier plants all around. This slide probably looks familiar to most of you, as it's a very common representation of issues related to crop production. Liebig's barrel, or law of the minimum, applies to agricultural production. It states that growth is dictated not by total resources available, but by the scarcest resource or the limiting factor. In short, you can have an abundance of all nutrients, but if you are missing vital micronutrients, for example, your crops may suffer and you will likely experience diminished returns. So balancing nutrients is very important in overall crop production and reduction of stress. When I was in school, we were taught about the 16 essential elements or nutrients related to plant nutrition. This graphic shows on the left side the 16 essential elements required by plants to grow and produce properly. The middle column shows the principal absorbed form of those nutrients, or the form that the mineral must be in for the plant to absorb it via the roots and then to utilize it within the plant. This form is a critical factor when making foliar applications of nutrients. You will notice that in most cases of the minerals, the form is in the cation form, meaning the mineral as being as a charged particle with nothing else attached to it. For example, calcium with a 2+ by it would be a calcium cation with 2 positive charges, or potassium with 1+, a potassium cation with 1 charge. The right side, or I should say the right column, shows the healthy plant concentration of the minerals that the plant requires for optimal growth and production. At the top of the list in that right column, we have what we call the primary nutrients: carbon, oxygen, hydrogen, nitrogen, phosphorus, and potassium. These are going to be the elements that are required in the highest concentration in the plant. Plants obtain the carbon, oxygen, and hydrogen from the atmosphere and water and rely primarily upon roots in the soil or other growth media for all of the rest of the nutrients listed on this graphic. The secondary nutrients are calcium, magnesium, and sulfur. The micronutrients are made up of iron, chlorine, copper, manganese, zinc, molybdenum, and boron. Note in the right column the concentration required by healthy plants. You'll note that it is listed as a percentage for the primary nutrients and the secondary nutrients, with the micronutrients being measured in parts per million. Just to give you a reference on the scale here, remember that your chemistry professor taught you that 1% is equal to 10,000 part per million. So you can do the calculations and see that the concentrations are much higher for the primary and secondary nutrients than they are for the micronutrients. This does not mean, however, that they are more important. They are just required in greater concentrations. This next slide shows the importance of nutrient balance. On the right-hand side of the scale, you can see the primary nutrients listed as N, P, and K, the secondary nutrients, calcium, magnesium, and sulfur, and then the micronutrients, zinc, iron, manganese, copper, boron, molybdenum, chlorine. All of these nutrients must be in balance in order for the plant to be able to produce effectively whatever it is being grown. If any one of these nutrients becomes out of balance, then that nutrient will likely become a or the limiting factor to production. Think back to Liebig's barrel and the law of the minimum. Let's talk about mineral movement within the plant. Minerals move by different mechanisms within the plant: acropital movement and basipital movement. In acropital movement, minerals move in 2 ways. Most common is from roots to leaves and or growing points. Uptake and movement from the roots require requires minerals to be in a form that plants can utilize. This type of movement utilizes the xylem pathway. Next is basipital movement. Another form of mineral movement is from from leaves to roots. This requires minerals to be soluble and able to penetrate leaves and move through the plant. This utilizes the phloem pathway. Once a mineral is in a plant, it must move from the stem to outer growing points via translocation. Many minerals have very limited mobility once in the plant. One way to look at mineral movement in the plant is to discuss the velocity of mineral movement. You can see on this graphic in the bottom of the slide the percentage or ability of minerals to move within a plant. Many necessary minerals have limited mobility in the plant. Even when minerals are in the soil in abundance, they can be unavailable to plants in their natural state or the state in which they are occurring in the soil. Minerals may also be bound in areas of the plant, which makes them highly immobile. Thus, minerals that the plant needs often cannot reach growing points where plants need them the most in a form that the plant can utilize. If we assume that nitrogen has 100% movement, this scale shows the relative ability or inability of the other nutrients to move in relation to nitrogen. Potassium moves at about 90%, so it's quite mobile. Then, looking at the 50% area, we see phosphorus, sulfur, magnesium, manganese, iron, zinc, copper, and molybdenum, all being 45 to 65% mobile. Then, decreasing to the left, boron is about 20%, and then the mineral with the least ability to move in a plant is calcium, which moves very slowly in a plant when compared to nitrogen. So next, let's talk about the foliar application of nutrients. There are many barriers to the absorption of foliar sprays, which result in limitations when applying minerals to the foliage of plants. The first barrier is the plant's cuticle. Another barrier is the plasma membrane. Molecule size can also be a barrier, as well as the chemical's electric charge and solubility. The plasma membrane only allows molecules that it recognizes as being naturally occurring, as well as they must be small enough to pass through. Remember the earlier slide that showed the principal absorbed form of minerals? Most of them were ions, anions or cations, positively or negatively charged. More about that on the next slide. Solubility is also critical. If a compound is soluble, let's use calcium chloride for example, the calcium chloride dissolves or dissociates when put into a spray tank, meaning the calcium is a positively charged cation and the chlorine is a negatively charged anion, and they are no longer attached to each other. the form of calcium, the form that the calcium is in at that point is the correct form for the plant to use. It's cationic form, being calcium with a +2 charge. Comparing that to calcium oxide, which is insoluble in water, the calcium does not convert to a form in the spray tank that the plant can use. It still remains bound to oxygen. The leaf's cuticle is a significant barrier to the absorption of foliar sprays. Cutin, which is found in the cuticle of the leaf, is made up largely of various combinations of members in two groups of fatty acids. Many waxes contain long-chain fatty acids. It is mainly these fatty acids which are responsible for the negative charge of the leaf. Now, recall in the previous slide where I discussed the positively charged cations. If positively charged cations are sprayed onto a negatively charged leaf surface, most of them become tied up on the surface of the leaf due to the opposite chemical charges. The cuticle also aids in keeping certain stressors out of the plant, such as fungi. It also keeps water in the plant. Mineral supplements can become trapped in the cuticle, and when we apply minerals to the foliage, they must enter the leaf to move to the growing points. They must pass through the cuticle and the plasma membrane and end up in the cytoplasm. When making foliar applications of nutrients, it is important to reach an ideal balance. Metallosate molecules do just this, as they have a small molecular size, they are neutral in charge, and the minerals are protected by amino acid chelation, which allows them to penetrate the cuticle of the leaf, pass through the plasma membrane, and ultimately reach the cytoplasm and be moved or translocated throughout the plant. Another thing just to note on this picture are the two water droplets in the slide. You'll notice that the water droplet on the left contains no surfactant compared to the water droplet on the right that does contain surfactant. Surface area is very important in foliar application of minerals. That water droplet on the left with no surfactant is actually being hung up on the waxy pillars of the cuticle, and it provides no surface area for the contents of that droplet to be absorbed into the plant. Whereas compared to the droplet on the right, there is a much greater area of that droplet contacting the cuticle of that leaf, allowing the opportunity for absorption of whatever is in that droplet into that leaf. So a surfactant is very important in the foliar application of nutrients. Translocated throughout the plant is where we prefer minerals to end up. For proper metabolization, minerals must cross the primary cell wall structure. The small neutral molecules contained in the metallosate products can penetrate the cell wall and move into the cytoplasm. Amino acids aid in the absorption of nutrients across the plasma membrane and also provide energy to the cells. Metallocate minerals are more effective than inorganic minerals at being metabolized in the cell. Moving through the phloem in many cases is from high to low mineral concentration. Movement can be in all directions. Nitrogen, phosphorus, potassium, and magnesium move relatively easily throughout the plant. Photosynthates, carbohydrates, sugars, proteins, enzymes, and hormones also move in the phloem system. Metallocate products act like naturally occurring chelates. The amino acid rings disguise the mineral to appear as a protein, making it mobile in the phloem system as well. Now a little bit on our Metallocate Stim Plus biostimulant product. Metallocate Stim Plus acts as a biostimulant that aids the plant in overcoming both biotic and abiotic stresses by utilizing a unique mode of action to increase the health and productivity of plants. Amino acids have been used in plant nutrition for over 5 decades to enhance growth and give an energy boost to a plant's physiological systems. Amino acids are the building blocks of protein, and as such are critical to the growth and development processes within plants. By supplementing the plant with additional amino acids, we provide the plant with more energy to grow. All plants require a balance of 16 essential minerals to grow properly. These minerals are major factors in everything from cell wall structure and synthesis to photosynthesis, protein synthesis, and enzyme activation. Metallocate Stim Plus contains a combination of several common micronutrients that work in an interconnected manner to enhance the productivity of the plant's many biological systems. Seaweed extract provides additional nutrients and energy in the form of natural auxin and cytokinin to plants. It can promote increased vigor, improve overall plant health, and greater productivity as it is applied to plants. It can cause increased root and vegetative growth by stimulating natural responses within plants when applied as a foliar spray. It can also have positive impacts on soil microbiota. Next in the discussion today, let's take a look at 12 different minerals, how they function within the plant, and some of the deficiency symptoms that result when the minerals are not present in high enough concentrations. Nitrogen. It is a component of amino acids, proteins, nucleotides, nucleic acids, chlorophyll, and coenzymes. Amino acids are the building blocks of protein, and they are what every living thing is made up of. There are 20 amino acids, and all 20 of those amino acids contain nitrogen. This is why plants require so much nitrogen to grow and develop properly. Chlorosis is the main deficiency symptom. Deficiency symptoms begin in lower leaves, which indicates nutrient mobility. Phosphorus. Phosphorus is a component of energy-carrying phosphate compounds, ATP and ADP. It has a key role in photosynthesis and respiration. It is mobile within the plant. Deficiency symptoms include dark green color, purplish leaves, and delayed maturity. Potassium. Potassium is involved in osmosis and ionic balance. It regulates the opening and closing of stomata. It's an activator of many enzymes, and it is mobile within the plant tissue. Deficiency symptoms include chlorosis, margin or tip burning on leaves. Within the plant, potassium functions in the translocation of sugars, manufacture of carbohydrates, promoting root growth, resistance against disease invasion, and in the overall quality of the crop. The next one is calcium. Calcium is the main constituent of cell walls in plants. It's very important in cell wall integrity. It is essential in maintaining stem strength, fruit firmness, regulating how cells absorb materials, including nutrients, across their membrane. Calcium is not translocated in the plant. Throughout my career, I have seen the importance of calcium applied to crops steadily increase. If you are producing an item where shelf life is critical to your success, calcium is very important to you. The longer something must be stored, or the farther it is shipped, the more important calcium becomes. If we consider the natural process of fruit rot, which is basically when the cell walls break down, the cells then leak out the content, and the fruit breaks down and begins to rot and decay. The deficiency symptoms include death of growing points, dropping of blossoms or buds, bitter pit in apples as shown in the slide, and blossom end rot on tomatoes. Magnesium. Magnesium is the central core element of the chlorophyll molecule. Magnesium is mobile within the plant. The deficiency symptoms are often chlorosis in the older parts of the plant. Because magnesium is mobile, the plant can take it from the old tissue and move it to young tissue, resulting in chlorosis of the older parts of the plant. Sulfur. Like nitrogen, sulfur is a constituent of protein. There are two amino acids that contain sulfur, cysteine and methionine. Chlorosis is the most common deficiency symptom. Not having enough sulfur limits protein production in the plant. Next is iron. Iron is essential in activating the enzyme system which builds the chlorophyll molecule. The lack of iron results in chlorosis. It can be very localized in a vineyard, orchard, or field. I have seen in orchards, for example, where one limb of a tree, all the leaves are yellow, with the rest of the tree having nice, healthy, green-looking leaves. This is not uncommon to see with iron chlorosis. It's also common to see areas or patches in fields or orchards or vineyards that are green with other areas that are very yellow with chlorosis, relating directly to the lack of iron. Manganese is very closely related to iron in its functions in the plant, and deficiency symptoms are often very similar. Manganese is required for integrity of chloroplast membrane and for oxygen release in photosynthesis. Next one is zinc. Zinc is essential in the activation of many enzyme systems within the plant. It is only very slightly mobile within the plant tissue. Deficiency symptoms can include decreased stem length, rosetting of terminal leaves, as shown below in that apple branch, and sometimes chlorosis. Oftentimes, it is expressed as well in the leaves, where the leaves are smaller and often very misshapen, commonly seen in grapes, for example. The next one is copper. Copper is essential in the activation of many enzyme systems. The deficiency symptoms include chlorosis and the dieback of terminal shoots. The last one is boron. Boron influences membrane integrity as well as calcium utilization. You can have adequate calcium in the crop, but without adequate boron, the plant is not able to utilize the calcium that is there. Boron is very important in the way plants use energy for growth. Boron is not mobile within the plant. Deficiency symptoms include death of terminal growth, improper flowering and pollination, and low fruit quality. Boron is often applied during or just prior to bloom periods on perennial crops. Cell growth and elongation is dependent on adequate levels of boron. This is especially important when discussing pollen tube growth and elongation during the pollination process. Proper pollination and fruit set is heavily dependent on having adequate boron levels in the crop. Thus, the reason for making applications of boron during the bloom period. Balanced nutrition leads to better stress fortification in plants. Healthy and balanced plants are more resistant to abiotic and biotic stress. Each nutrient plays a vital role in keeping plants balanced and allowing plants to thrive. Plants must maintain a delicate balance of nutrients to perform many physiological processes simultaneously. Minerals, seaweed extract, and amino acids can provide support to plant systems in times of stress. Supplementing plants with the correct nutrients that are bioavailable and can move to growing points is a key to fortifying plants against stress. Okay, so that makes up the bulk of what I had wanted to present. So I think we'll maybe look at some questions.
All right, thank you, Jeremy. We do have time for questions, and I'd like to encourage you to go ahead and submit some if you haven't already. But we've gotten quite a few in, so I'm going to get started with those. The first question is, and it's kind of a broad one, what are the most important micronutrients?
Yes, okay, and that's a great question. And I would say there's probably two parts of that answer. The first one is, I would say the most important micronutrient is the one that you're lacking in your crop or in your production system. And the reason that is that's going to be your limiting factor. And so if you're growing, let's say, apples, and you don't have adequate calcium, that's going to be your most important nutrient to apply. But it really depends on what you're low on, because they're all important in the overall production. That balance that I talked about earlier is very critical to have optimal production. So the most important one is the one that you're lacking, I would say.
And I imagine it depends on the crop phase too, right? Different crops require different nutrients at different times in production. Is that correct?
You bet. That is absolutely correct. Yeah, there are different growth stages that have a much higher demand for the nutrients. For example, on grapes, when they're going through veraison, potassium is very important to allow those sugars to be translocated into the berries.
Mm-hmm. All right, we'll move on to the next question, which is, I understand that citrus trees have a waxy coating on the leaves, which doesn't allow for good absorption of foliar spray products. What to do? Yeah,
that's a great question as well. And some crops have a much more waxy and a thicker cuticle than other crops, and so that's where the formulation of the mineral that you're applying becomes very, very important. Of course, a surfactant, which I already mentioned, goes without saying. Surfactant should be mixed with any foliar application to be applied. But the form of the mineral is very important. If you're applying just a calcium chloride, for example, where you've got calcium cations, they're going to really be tied up in that cuticle. So the product chemistry or formulation is very important in allowing the minerals to move through that very waxy cuticle on that leaf. Another example of a very waxy cuticle is like on broccoli, for example. Very waxy, and that water droplet will just run right off of that unless it's having a surfactant in there. So.
All right, sounds good. Our next question is, how do we determine nutrient deficiency? I mean, you mentioned the symptoms, the visual symptoms on the leaves. Is there any other way? Do you recommend tissue testing, things like that?
Yeah, that's a great question and a great point. So I've always been a strong believer that if you're observing a visual deficiency, the cow is out of the barn and halfway onto the neighbor's field, and you've got a lot of catch up to do to remedy that deficiency situation. So what I promote and think is the best way to determine the overall nutritional status of your crop is tissue analysis. So there are different sampling times for different crops and different methods, but certainly a tissue analysis is a very good indicator of what's happening at any given time in that crop, and you'll pick up a lot sooner on that method of a deficiency rather than relying on a visual symptom. If you're observing deficiencies, then you've gone down the path pretty far, and it's hard to get back to where you need to be with appropriate nutrition. So yeah, absolutely, plant tissue testing is essential, and not only that, but soil testing is another very important tool that must be used in your production system. You know, soil sampling is done once, sometimes every 2 years, but it's a gauge on what's happening in the soil, what's available, what the pH is, and you can determine what nutrients are there, what are not there. And you can also use that as a gauge, you know, to determine, looking at soil analysis and plant tissue analysis, and see how effective your current fertility program is. If you do a tissue analysis and find that your let's just say that your phosphorus levels are extremely high in your tissue, then you can make an adjustment to your soil applied fertilizer program and not be putting on as much phosphorus, or make amendments to adjust whatever is happening. So plant tissue analysis is a very, very important tool in engaging all of those things.
All right. Our next question is, what is the optimum molecule size for foliar applications?
That's a good question. If you look at molecule size can be measured in Daltons, and that's basically a mass measurement. They typically, rule of thumb is 1,000 Daltons. If you're bigger than that, you're too big to be absorbed through the plasma membrane. So the target is 1,000 Daltons or less, and that's actually a measurement of mass.
And I'm guessing that the foliar product that you have, the metallosate or metallosate, I'm sorry, I should pronounce it correctly.
That's okay.
Is that the right size?
Yeah, absolutely. That's a great point. Yeah, the metallosate molecules are very small, in the range of, a couple hundred Daltons in size versus, something that, like a long chain protein or, something of that nature that's much, much heavier, much larger.
Okay. All right, our next question is, how does a product like metallosate StimPlus protect or fortify plants in times of stress?
Yeah, so the combination of the chelated minerals, the amino acids, and the seaweed extract, all of those things are going to contribute to the overall health of that plant that you're applying it to. So, it's basically, the healthier the plant, the more robust it is, the better root system it has, the better leaf size it has, the better it's going to be able to resist or overcome, these moments of stresses, whether they're, biotic or abiotic. It just allows it to be better. It's similar to saying, if a person is, healthy and eating right and exercising, they're more able to fight off, a cold or an illness. And so it's along those same lines. And so that StimPlus provides that extra support to those plants that allow them to resist or to overcome those stressors.
Okay, this is a good question here. What happens to a mineral which gets absorbed by the plant, but it isn't needed by the plant at that point in time? Can it be stored and used later when needed?
Yeah, so that's a great question, and it's a little bit of a two-edged sword, because sometimes we can apply a nutrient to a plant, the plant doesn't need it, and that nutrient will become excessive and can throw that crop out of balance. Remember, that balance is what's so important, and we want that balance to be maintained. And so, yes, there is the ability of plants to store nutrients and store minerals, but the key is to, if you have a nutritional need, to only be applying what that crop needs, not other things in addition, because then you start throwing the balance of the nutrition out of whack in that plant.
Okay, and probably spending unnecessary money as well, I'm assuming.
Yes, exactly.
Okay, let's see. Do your products stay mostly in the cyan, or how much goes to the roots and then back up? That's our next question.
Let's see, trying to understand that one. So I would say the
Maybe they
cyan.
shoot.
So I'm going to assume they're meaning the top, the upper part of the tree. So
yeah, I'm
you know you got the rootstock and the cyan. So the cyan is ultimately, where that mineral is needed. The roots are more of the absorptive areas where the minerals are coming through. So while we do see movement, and we have measured that movement down to the roots using isotopes, typically most of it is going to stay, up in the cyan or the top, above ground parts of the tree in this case, and it can be, I would say, translocated or moved to the root systems if, for example, you're on a deciduous crop and you're trying to, get stored up for the winter. You're moving, some carbohydrates and photosynthates down to the roots. It can move that way, but typically, with the metallocene products, we see that it's going to stay in the, in the above ground parts of the crop, put it that way, where it's going to be utilized.
Okay. Let's see. I've got a few more questions here. There are a lot of foliar nutrition options. Why should I use metallocene?
There are a lot of options, and I would say, my recommendation is use a product that you're going to get the full benefit out of the minerals that you're applying. So with metallocene, you're applying an amino acid chelated mineral where all of that mineral is going to get into the plant. It's not going to get tied up on the leaf. It's not going to wash off. It will get whatever is sprayed onto the leaf is going to move through the cuticle, through the plasma membrane, and then into the cytoplasm, and then be utilized by the plant. So that's my recommendation. Use something that works and something that is safe, that you can see the benefits from what you're applying.
Okay. All right. Our next question is how often or frequently to apply foliar spray products to stressed trees? So, how often should you apply foliar sprays to stressed trees, I guess is the question.
I mean, it's kind of a relative question. It depends on how severe the deficiency is or what the stressor is, what's happening. So there's not really a solid answer. I could say every week or every 5 days or 10 days. It kind of depends on what the problem is and how you're trying to what you're trying to address. So yeah, that one's not a clear cut answer on that one.
Okay. One of our viewers is asking why you didn't mention soil pH in terms of how it affects the use of nutrients in the soil and within the plants. Maybe you could talk about that a little bit. I know that's a big subject, but
yeah, no, it's a great question, and I appreciate that question. Looks like it's from Tom. Soil pH is obviously a huge player on plant nutrition and plays a key role. That's why you should be taking those soil samples, as I had mentioned, to see what the pH is and be able to determine what amendments need to be made to get that pH ideal for whatever the crop you're growing is. For example, if you're growing blueberries, you want that pH to be down around 5. Most crops, prefer a more neutral range of pH. And so, yeah, it is very key, and I'm sure you can all remember that funny graph with the bars that go across, and they get fat and thin as the pH changes on the bottom from acidic to alkaline. And so, yeah, it is a big player in nutrition. I didn't go over it in depth on this webinar because I wasn't really focusing on soil. However, yeah, it's a big part of the puzzle, no doubt about it, and something that should always be addressed and observed and looked at constantly.
And this is a question for me, I guess, following up. Is I know you usually determine the soil pH. You have to do a soil test. And we talked about soil tests earlier too. So what time of year in the season would you recommend doing a soil test? Like after harvest, before planting? What would you recommend?
Yeah, so it depends on the crop, and it depends on the climatic conditions of where you are, because rainfall can have a big impact on pH as well. But typically growers, at least let's say in North America, they're going to test usually in the late summer or fall, and once that's been started, then you kind of use that as a baseline and sample at a relatively similar time every year, so that your data becomes more accurate, because if you're sampling in the spring and then the fall, and then you decide, well, I want to look at it, in the summer next time, things really can change throughout the season. So whatever the time is, I just say stick with that timing and build your knowledge database on a set timing. But it's usually, in the fall, late summer or fall, at least in the northern hemisphere. Other areas where it's temperate or tropical crops where you don't have the seasonality we do, then it's just, pick a time and just be consistent.
Okay. All right, we had another question come in. How quickly do the metallicate products move through the plant compared to basic sulfate-based foliars? Yeah,
so that's a good question. So if you're looking at just the basic soluble salt fertilizers like a zinc sulfate or a iron sulfate compared to metallocate, those sulfate fertilizers, majority of those are going to be tied up on the leaf, so they're not even going to get into the plant. And so the metallocate products, they're going to be absorbed through the cuticle plasma membrane and into the plant within 2 to 3 hours after application. So you make your application in the morning, by midday, it's not on the leaf. It's in the plant. it's actively working and doing whatever the plant needs it to do. So it's very quick with these products because of the chemistry involved in the formulation.
All right. Well, that is the last of our questions. So I want to thank you, Jeremy, for sharing your wealth of knowledge about agronomy. I'm sure you could probably do several presentations given your experience, so thank you. And as a reminder to our audience, you can go back and rewatch parts or all of this webinar at the same link that you used to get on today. And you also get a reminder tomorrow, with that link as well. So if you want to go back and check any facts, you can do that. And you can also find the link at growingproduce.com/webinars. So thank you so much for joining us today, and thank you to Balchem Plant Nutrition for making this webinar possible. So have a great day.